Hi everyone! After my last video, I received a couple of questions about this new strain of COVID, as well as what happens after we have vaccine rollout. To address both of these questions, I think we could actually talk about testing. What types there are, how it's done, and why testing is as important now as it was at the beginning of the pandemic. There are three main types of COVID tests available: molecular or PCR, antigen, and antibody. These tests all look at different parts of the viral infection, so let's start with a brief overview of how viruses work. There are many different types of viruses, but viruses like COVID carry a copy of RNA, which acts like the instructions for building more virus, just like the RNA in humans carries the instructions for building a human. The goal of the virus is to break into your cells and use the factories your cell uses to make things for your body, instead to make pieces of the virus that can be assembled, therefore replicating the virus. In fact, the virus is technically not a living thing. It's like a very good hijacker that takes advantage of the body's efficient factories to do all of the work for it. So here's where we run into our first type of test: molecular or PCR testing. This test functions to look for the RNA instructions for building a virus and see if it's inside your body. Since viruses don't really look like human molecules, it's fairly easy to spot the difference between human RNA instructions that are supposed to be there and virus RNA instructions that aren't. Scientists do this by taking a sample either ideally from your nose or possibly from your throat or mouth and using the equivalent of a molecular copy machine to make a ton of copies of the viral RNA instructions. The catch is that if there aren't any instructions, there's nothing to make copies of, and you'll get a negative result in 1 to 7 days. If there are RNA instructions, the scientists will see that there are a ton of copies, and that means that you're currently infected. This test is the most accurate way to determine if you are currently infected with the coronavirus, but this depends on timing. Imagine if you got tested right after you were exposed. There are probably only a couple of copies of the virus RNA instructions in your body, which makes the odds that those very few copies make it into that sample taken when there are trillions of cells in your body very low. That's why it's important to take the test after you've started experiencing symptoms or more than 7 days after potential exposure. This makes sure that the virus, if present, has plenty of time to make copies that we can detect, therefore giving you a more accurate result. That's why even after a negative test, it's important to quarantine and avoid others as much as possible, just in case you were tested too early or too late. So this test does tell you if you are currently infected with COVID. What this test does not tell you is if you have previously been infected or if you are immune to the virus in the future. All it tells you is if you are infected with a detectable amount of COVID right now. So let's go back to our discussion of how viruses are detected by the immune system. Basically, our immune system employs cells that act like security guards to patrol the body and point out anything that looks suspicious. These things that look suspicious are called antigens. There are a lot of these on the virus, but the virus's job is to try and make these things look as inconspicuous as possible so the security guard cells won't know anything is wrong. This means that sometimes these antigens can look like other things that are supposed to be in the body, which is an idea that we're going to come back to. This brings us to our next type of test, the antigen test. In this test, a technician will take a sample from your nose or throat, mix it with some chemicals to cause the virus to let go of many of its pieces, and then place this mixture on a special type of paper that recognizes viral antigens and will change color once bound. Because this test is much simpler, this means that it can often be returned in an hour or less. But this comes at the expense of accuracy. Remember how I said that sometimes virus antibodies try to disguise themselves as pieces of human? This makes it really difficult for the test to tell the difference, which leads to false positives, which is being told you're infected when you aren't, and even more importantly, false negatives, being told you don't have the disease when you do. Because there is a much greater chance for error with this test, it's important that you quarantine and remain vigilant even after a negative test. And, just like the molecular or PCR test, it is best to take this test more than 7 days after exposure or after you start experiencing symptoms. Finally, this test does tell you if you're infected, but it does not tell you if you are immune or if you were previously infected. Let's revisit the immune system and its response to viruses one more time. Once the security guard cells identify a potential threat, they start a big response. But this response can't just start killing cells all willy-nilly, it has to be targeted. One way to target the virus and infected cells but not healthy ones is by using antibodies. These are like unmanned drones that are designed to fit perfectly with pieces of the virus and block it from being able to continue to infect you. It usually takes a while for the body to find this perfect fit, which is why you'll start to get sick as the virus continues to gain ground. Once your body has developed the perfect antibody drone, it can reduce the number of effective viruses and help you start to heal. This is the idea behind convalescent plasma treatments that you may have heard about. If we take antibodies out of one person who already has them and put them into another person, the drone antibodies can still identify the virus and help push it down. The success of this treatment depends on a lot of factors, however, and is very expensive. These drones usually hang out for a while, even after the infection has completed, to make sure that no more of that virus is trying to sneak back in. This leads us to our last type of test, the antibody test. This test is performed by taking a blood sample and looking for your body's antibody drones in that sample. If your body has drones out and patrolling, they'll be able to be detected, which tells scientists and technicians that you, at one point, were infected with the virus. However, this test cannot tell you if you're currently infected. It also does not tell you if you are immune to the disease. There is a ton of research going on in this area, but as of right now, there's no evidence that antibodies will prevent reinfection. These drones may start to decrease after a certain amount of time, they might not be able to recognize a virus wearing a new antigen disguise, or they may not be enough to prevent the virus from infecting cells even while the drones are fighting the virus off, making you potentially asymptomatic but highly contagious. So what does this have to do with the new strain? This new strain is highly infectious, which means that it's made a couple of upgrades to itself to allow it to travel better person to person. These changes are reflected in its RNA instructions and, therefore, can be detected and traced using PCR and molecular tests. However, at its core, the virus can't change everything about itself, which is why many of the antigens it presents are the same and why antibodies seem to work on both. Again, this is all new, and as we get more information, we'll know more about what this new strain means. But in the meantime, it means to continue to stay home, stay vigilant, wash your hands, wear a mask, and look forward to the vaccine. Stay safe, everyone.